Good morning, kids. Hope you all are enjoying the lectures. I think that you have all got a pretty good idea now regarding how light is reflected, refracted, how images are formed by a spherical mirror, by lenses, right? So now, when we are learning all these things, about the things which are there outside, okay, why not study our own eye, right? Our own mysterious human eye. How are we able to see all the things? How are we able to perceive all the things, right? Human eye is just like a camera, okay? It just clicks on whatever we see. It takes an image and imprints on the screen. It takes an image, imprints on the screen, right? So when we see continuous form of these images, we see what is going there in front of us, right? So let us see how is the structure of human eye. How does the human eye actually work, right? Let us start. Now, you know how do we perceive objects, okay? So this bulb is the source of the light. Light rays from bulb, right? Light rays from bulb fall on this ball. This ball will reflect these light rays and it will reach eye. Because of this, because of these light rays reaching our eye, we are able to perceive the ball. Okay, if there was no light, there was no light rays, we are not able to see anything. Can you see anything in a dark room? No, we are not able to see anything in a dark room because there are no light rays reaching our eyes, right? Okay. Now, as far as the different structures or the different parts of human eye is concerned, we will go step by step, one by one. But before that, I'll just give you an idea. What does the human eye actually consists of? The human eye has a cornea. Okay. What's the cornea? Cornea is this part. This is cornea. Okay. The first transparent layer that we can touch with our fingers. Okay. That is your cornea. Second, what you have? Second is your iris. Iris is this colored part. Okay. So for example, like you can see this person is having blue colored eyes. So this is iris. Inside the iris, you have this black eyeball, which is always there. So this black eyeball is called as pupil. This is the pupil, right? Then we have a crystalline lens. Can you see this transparent jelly-like substance? This is a crystalline lens. And in a human eye, the crystalline lens is always a convex type of a lens. A convex type of a lens is a converging lens. Okay, pupil, crystalline lens. Then we have ciliary muscles. Can you see this part? Okay, so this is your ciliary muscles. So these ciliary muscles hold the eye lens together. It holds the lens. I'll tell you why holding is required for the eye lens. This is the ciliary muscles. We'll study step by step each and every structure in detail. Okay. Then you have your retina. What is a retina? A retina is nothing but a light sensitive screen. This is your retina. All the light which is entering the human eye, okay, it will go through the retina, through the lens, and these rays converge on the retina, and hence we are able to see objects, right? If retina wasn't there, retina is your light sensitive screen. It is like you have a screen for a projector. Unless and until the screen is there, you are not able to see the images. So retina acts as a screen, okay, to view the images. After retina, on retina, you have something called as light sensitive cells. So as soon as these light rays come on the retina, these light sensitive cells get activated and they will send electrical signals to via the optic nerve to the brain, and then brain will do all the interpretation, and then we are able to see things which are around us. Okay, let us see in detail now, if you see your eyeball, basically only 30% of the eye is visible to you. 70% of the eye is inside what you can see inside the skull. So you are able to see only this part of the eye, but this much, this entire thing is there behind the eye. So an eyeball is nothing but a big spherical shape of radius 2 point of diameter 2.3 centimeters, right? It's spherical in shape. The first and the foremost thing that we can see or we can touch with our hand, that is called as cornea. Okay, cornea is a transparent bulge which is there in front of the eye. Light enters through a thin membrane that is called as cornea. So whatever light which is coming from the atmosphere, it has to pass through the cornea. Okay, and we say that maximum amount of refraction of light, maximum amount of refraction of light is only happening at the cornea. So this is your cornea. These are the light rays which are coming, and these light rays, because light rays now are changing medium, it is coming from air medium to cornea medium, then again to iris and inside the eyeball. Okay, so when it changes medium, we say that refraction is taking place. So 90% of the refraction which takes place is taking place at the cornea. Right? It forms a transparent bulge on the front of the surface of the eyeball. Most of the refraction of the light rays entering the eye occurs at the cornea. As I told you, this is your cornea. This is the cornea. This is the transparent one. After cornea, you have, this is your iris. Okay. After iris, this is the eye lens. And this dark thing which is there inside, this is pupil. We'll study all this in detail. Right? Understood what is cornea? Behind the cornea, we have iris. Okay. So this colored part, if you see this dark muscular diaphragm, this is your iris. Okay. In between the iris is your pupil. What is iris? Iris is a dark muscular diaphragm that controls the size of pupil. See, pupil is an inlet for light rays. Entire light enters the retina or enters the human eye through pupil. Okay. It regulates and controls the amount of light entering the eye. Now, if somebody just takes a torch, puts it on and brings it near to your eyes, you are not able to accommodate so much of light in your eye. So what you do, you just close your eyes for some time, or when you observe the pupil of that eye, the pupil size of the pupil just goes small. So if the size of the pupil goes small, that means light will be, so if so much amount of light is entering this eye, this much amount of light will only enter the retina. Okay. Now, when you go to a dim place, when there is not much light, when you go to a dim place, when there's not much light, pupil will relax and it will allow large number of light rays to enter. Because the light is dim, there are actually no light rays. I mean, very few light rays in that particular medium and the particular room. So the size of the pupil will enlarge. So this contraction, enlarging, means the pupil is relaxed. Okay. And becoming small means it is contracted. So this contraction and relaxation of pupil, who does this? Iris does this. So what is iris? Iris is a muscle. It's a dark muscular diaphragm that controls the size of pupil, right? Now, you must have observed that people have different colors of eye. Some people have blue eyes, some have greenish eyes, some have brown, some have black, right? So now this color differentiation, it is not the eye, it is basically the color of iris. Okay, color of iris can change, but the pupil will always remain black. Color of pupil never changes, right? Color of iris changes. So it is basically this blue colored eye and green colored eye. All that is differentiation because of the color of the iris, and that's basically genetic. Okay. Right. This transparent bulge, cornea. Then we have iris, the muscular diaphragm, which is holding the pupil together. What does pupil? Iris controls the size of pupil. In return, pupil controls the amount of light entering the human eye. Understood? Okay. See, as I had told you, a relaxed pupil. Relaxed pupil as in there's a normal amount of light which is entering the human eye. So in this time, see, if you see, the pupil is enlarged, so that means it is in a relaxed position and it is allowing maximum amount of light to enter. This basically happens when you go into a dim room, right? If there is, or basically during night. Okay. Now in the next slide we will see, now this is a contracted pupil. Now contracted pupil means large amount of light is entering. Okay, large amount of light. So sometimes when you go directly outside the house in afternoon where the sun is shining brightly, your eyes are not able to accommodate so much amount of light. So what does the pupil do? Pupil reduces the size. It becomes contracted and it will allow very less amount of light to enter the human eye, because if large amount of light enters the eye might get damaged. So this controlling of the amount of light is the work of pupil, right? So if you observe, see, small, big, small, big. So this is done by pupil. Who is holding the pupil? Who is controlling the size of pupil? Iris is controlling the size of pupil. So then next important part what we have is lens. Number one, what did we study? Number one, we had cornea, transparent outside bulge. Most of the refraction of light takes place at the cornea. Second we did was iris. Iris was the muscular diaphragm which holds the pupil and it controls the size of pupil. Third was pupil. Pupil, by contracting and relaxing, controls the amount of light entering the eye. Now the whatever amount of light which is entering the eye, now it goes to the lens. Now this is your number 4, very important. That is your lens, right? A lens, which type of lens this is? This lens is a convex lens. It's a converging lens, right? And if you see, this lens is holded together by these muscles. These muscles are called as ciliary muscles, right? So the ciliary muscles are holding our crystalline transparent lens. Now, a crystalline convex lens, what does the lens do? Because if you say most of the lens is a different medium, so obviously we say when light goes from air to lens, it will get refracted. But already when the light ray enters a human eye, change of medium is experienced by it. So already most of the refraction is taking place at this interface itself. Okay, it's taking place at this interface, that is the cornea. Now, what does the lens do? The lens does the finer adjustments of focal length, because we know a lens can be a thin lens, okay, or it can be a thick lens. So if I say this can be a thin lens, this is a thin lens, and it can be a thick lens as well, like this. A thin lens will have a larger focal length. A thick lens will have a smaller focal length. What is the significance of this? I'll tell you. Just wait for a minute. Okay. So first of all, just keep in mind that what is a lens? A lens is a crystalline convex lens. What does it do? It provides finer adjustments of the focal length. Why these finer adjustments of focal length is required? To focus objects at different distances on different retina. Now imagine when you just look here and there. In front of you, you have a book. You are able to see it clearly. Just a little beside, you have a chair. You are able to see it clearly. A little more further, you have a cupboard, a sofa, or a bed. You are able to see it clearly. Okay, irrespective of the distance of the object, you are able to see each and every object clearly, right? Unless and until you don't have a defect of vision. Okay, for a normal human eye, we are able to see each and every object clearly. But if you have done the image formations by lens very, I mean, by paying attention, if you have drawn the ray diagram for yourself, it depends on the distance of the object, where is the image formed, right? So by adjusting this focal length, we are able to focus objects at different places, and we are able to see them clearly. Again, a detailed explanation I'm going to give you now. As I told you, we have ciliary muscles. What does ciliary muscles do? These ciliary muscles are holding the eye together, holding the eye lens together, basically, right? So this is the ciliary muscle, which is holding the eye lens together. It's a fibrous, what again, this crystalline jelly, it's a fibrous jelly-like material whose curvature can be modified. Curvature can be modified, that means it can adjust its focal length. Who helps in adjusting the focal length? The ciliary muscles, which is holding the lens. It helps it in adjusting the focal length. Now, what does the eye lens do? When light rays, okay, when light rays from outside go through the eye lens, it's a converging lens, it will make a image. Where does the image will be made? The image will be made on the retina, right? If this is, so as these light rays are getting converged at this point, this is where your image will be formed. And how will be the image? As it is a convex lens, the image formed would be a real and an inverted image. Where is this image formed? This image is formed on the retina. What is retina? Retina is your light sensitive screen. Okay. Now, see if you can observe, this is the flower pot which was over here. Okay. Rays, straight rays from this flower pot, light rays went into the human eye. They got converged over here, and if you see, a real and inverted image of the object is formed on the retina. Now, as light falls on retina, what happens? So when this light rays get converged at retina, we know an inverted image is formed, real and inverted image is formed on the retina. Okay, what it actually do? It activates the light sensitive cells. Which are the light sensitive cells? Rods and cones. Okay, if you see, rods and cones are your light sensitive cells. If you can see, as soon as light touches the retina, these light sensitive cells get activated. Rod cells are activated only in dim light. Okay, tell me, you are sleeping at night. There's no light on in your room, and you want to take in dark only. You want to just observe something. Will you be able to observe that object very clearly? Will you be able to differentiate its color very clearly? No, because the light is dim, right? Now, rod cells get activated only in dim light. Okay, so that is why they are not able to differentiate between colors. They are not color sensitive, because in dark, when low light is there, dim light is there, we are not able to differentiate between different types of colors. Okay, on the contrary, we have another type of light sensitive cells that is called as cones. What are cones? Now, these cells are activated in bright light. Okay, when the light is more, cone cells get activated only in bright light. Hence, these cone cells can differentiate between different colors. So they are color sensitive. Cones, C for cones, C for colors. So cone cells are color sensitive because they get activated in bright light. Rods are activated in dim light, hence they cannot differentiate between colors. Okay. Now, people who have color blindness, right? People who have color blindness, they have some defect in their cone cells. Okay. Now, as these cells get activated, what happens from the human eye? They will send these electrical signals to the brain. Now, how is this electrical signal sent to the brain? It is sent to the brain via the optic nerve. Okay, this is the optic nerve which sends these electrical signals to the brain, and these get activated. Now inside the brain, now if you have been going through this lecture properly, you would have, a question would have arrived in your mind that if the object is rect, a real and inverted image is formed on the retina. So when these cells get activated, it goes to the brain and the brain interprets now. So do we see everything inverted? Because the image formed on the retina was inverted, right? So whatever we are seeing in front of us, is this inverted? No, it is the way it is. Now, this is the beauty of our brain. What does the brain do? The brain interprets these signals and finally processes the information that we perceive these objects as they are. So when these light sensitive cells sends electrical signals to the brain perceives these signals, and the brain itself inverts the images again. Now, when it inverts the images, we can see erect images or upright images and can perceive just the way they are, right? Understood? Of the same size, same shape, same color, right? So this is the beauty of our brain. Now, just in a nutshell, let us see what all we did. So 7 to 8 things that you have to put it in your answer. If a 5-mark question comes, explain the structure of human eye along with a diagram. Okay. I will have one more tutorial which is based on step by step how to make a structure of a human eye. Okay, so you can refer that tutorial for drawing a human eye, how a human eye is drawn, because this question is important. Normally comes for 5 marks to explain the structure along with the diagram. So the first and the foremost thing that you have to add is number 1 is cornea. What is cornea? Cornea is a transparent bulge. It is a white and transparent material. The first layer, it refracts the light, protects from macula, doesn't allow foreign particles inside, because if foreign particles are try to enter our eye, we close our eyes and a liquid sort of comes into our eye, which we call it as tears. Okay, so with the help of these tears, these foreign particles can just go out. So the number 1 we have, that is cornea. Number 2, the next most important thing, we have iris, which determines the color of the eye. It controls the amount of light entering in the pupil, or we can say it controls the size of the pupil. The pupil in turn will control the amount of light. Okay. Next we have is our convex lens, a crystalline jelly-like material. It's a converging lens. It will converge the light rays which is coming from the air medium and put it in and focus all these light rays on the retina. Number 5, what do we have? Now these, again, one more thing you have to add over here. These lenses, convex lens is held together with the help of ciliary muscles. Ciliary muscles, right? Then we have number 6, that is, what do we have? We have number 6, that is your retina. Okay, what is retina? Retina is a light sensitive cell. This light sensitive cell gets activated whenever light rays are coming on it, or get focused on it. These light sensors, after getting activated, they will send electrical signals to the brain via the optic nerve. So these signals are sent via optic nerve, at the last will go to the brain. Now, which are the 2 types of light sensitive cells? Rods and cones. What are rods? Rods are sensitive to dim light. Cones are sensitive to bright light. Hence, color differentiation will be there. Cones will be able, because of the presence of cone cells, we are able to differentiate between different colors. Okay, rods cannot differentiate between different colors. So, in a nutshell, this is what we have. Right? In a nutshell, these are the different types of structures, or these are the 8 points that you have to add while answering a question on describe the structure of human eye. Okay. Now, how is light propagated inside the eye? If you see, this is an air medium. Okay. The first, we say that if some foreign particle comes in, there is a lot of liquid which comes in. So, what is this liquid? This liquid is called as aqueous humor. Okay. This is aqueous humor. The second type of liquid which is over here, that is called as vitreous humor. Vitreous humor is there over here. This is your retina, this is your biconvex lens, this is the cornea. So just light enters, gets refracted at the cornea, little more refraction through the lens, and this is how we get an image on the retina. Now, why does it say that most of the refraction is occurring at cornea? Because if you see, what is the refractive index of air? It's 1. Refractive, on the contrary, when you compare, refractive index of cornea is 1.376. So if you compare the refractive index of cornea with aqueous humor, 1.3636, biconvex lens, refractive index is 1.4, vitreous humor, refractive index is 1.337, and then finally it's the retina. So out of this 4, the difference between the refractive index of air and cornea is the highest. So that's why most amount of diffraction is going to take place at the cornea. Now, on the contrary, the other 3 mediums which the light rays goes through, that is the aqueous humor, then you have the biconvex lens, then you have the vitreous humor. All these 3, 1, 2, and 3, all these 3, the light rays will both pass through this, but there wouldn't be much more refraction, because there won't be much more refraction because if you observe the refractive indices, there is not much a difference in that, right? 1.336, 1.4, 1.337. So little refraction is going to take place over here. But most amount of refraction takes place at the interface of air and cornea, because the difference in the refractive index is the highest, 1 and 1.37. Now, focusing on far away objects, lens becomes thin. Okay, when focal length increases, now when we have to just consider a scenario, when we have to focus light rays far away, okay, far away from us, what will happen? Light rays will come parallel. As you can see, this flower pot, these light rays are coming parallel. So in order to focus on far away objects, what will happen? Our eye lens becomes thin. Now, if this is a thin eye lens, so it is a part of a thin curvature, and as the circle increases, your radius of curvature increases, and hence focal length increases, right? So when we have to focus on faraway objects, remember the eye lens becomes thin. As the eye lens becomes thin, focal length increases. On the contrary, when we have to focus on nearby objects, the eye lens becomes thick. Can you see this? The eye lens is becoming thick. So if this is a thick lens, if you observe, it is a part of this, your focal length decreases. Okay. Nearby objects, focal length decreases, the eye lens is becoming thick. If you see, the eye lens is thin, the focal length over here decreases because the object is nearby, the eye lens is thick. Okay, now this ability of the eye lens to adjust its focal length is called as power of accommodation, right? Now this is what we called as power of accommodation. And so what does it do? So our lens sometimes thin, sometimes thick, sometimes thin, sometimes thick, depending on what type of object we are focusing upon. And this is a continuous phenomenon, because all the things that we see around us are at different distances. So each and every moment, the eye lens is adjusting. These finer adjustments of the focal length are done. And who helps the eye lens in adjusting its focal length? It is these ciliary muscles. Okay, these ciliary muscles, it will stretch the eye lens, it will relax the eye lens. Stretch the eye lens, relax the eye lens. Okay, so that is why your ciliary muscles are also important, because the ability of this eye lens to adjust the focal length. Okay, now this entire phenomena, that this ability of the eye lens to adjust the focal length according to the object, is called as power of accommodation. Right? Now when this object is very close to the eye lens, we cannot see clear images. For example, if you just take a book very, very, very close to your eye, okay? If you take a book very close to your eye, you are not able to see what is written in that textbook. Why? Because it is very near to your eye, and when it becomes very near to the eye, the images are formed over here. It is behind the retina. When the images are formed behind the retina, what you will see will be very unclear and blurry. Because, okay, basically it will be very blurry. So now when this becomes blurry, you cannot read it very clearly, right? So that is why there is a minimum distance. So we strain our eyes, the focal length of the convex lens decreases because it becomes bulged, right? So then there is a minimum distance at which distance we are able to focus the light rays properly so that the object becomes clear. Now this distance is called as minimum distance of distinct vision. So for a human eye, that is 25 centimeters. So this 25 centimeters is the minimum distance at which the human eye is able to see objects clearly. Okay, so always remember, for a normal human eye, we can see objects clearly up to a distance of 25 centimeters and not less than that. If we just hold this book here at a distance of 15 centimeters, proper images are not formed, blurry images are formed, right? In the same way, we have something called as, okay, so minimum distance at which objects can be seen most distinctly without strain is called as least distance of distinct vision. Very important, can come for a 1-mark question. It is also called as the near point of the eye. So for a normal young adult with normal vision, for a normal eye without any defect, the near point of the eye is about 25 centimeters. Now, as we have a near point, we also have a farthest point. The farthest point where we can see the objects very clearly. So for a normal human eye without any defects, the farthest point will be infinity. Okay, right, fine. Hope you have understood the entire structure of human eye. What all points you have to add to the structure of human eye? First, we have cornea, then we have iris, pupil, eye lens. With eye lens, we need to add ciliary muscles. Then we have retina. After retina, light sensitive cells, electrical signals, light sensitive cells, rods and cones, electrical signals via optic nerve to the brain. Right? So these are the 8 to 10 points that you have to include when a 5-mark question is asked about the working and the structure of human eye. Right? In the next tutorial, I am going to show you how to make a proper human eye with easy steps, right? Step-by-step tutorial I'll tell you. Next class, we will also do certain refractive defects of distinct vision. So you must have seen sometimes people are not able to see it clearly. So that's why they wear spectacles. Now, why are these spectacles? Why are they have to wear the spectacles? And why are they not able to focus properly or see objects very clearly? Okay, so 5 types of refractive defects we are going to study in the next class. Thank you. Bye-bye. See you soon.